What have you?
You didn't even get any. This is why are you recording this? I got 2.
But I don't think Dash and Ford Apple, they didn't get any photos.
Yeah, genius. Look at that.
I didn't spoil your
If I can see
filling up my hair, quite literally, to use as a DNA sample, and then so you've got to pull out your hair and cut the root with a little bit of hair on it to be used in the PCR.
Yeah, so then we don't need that bit.
3
Part. What you need to do is hold the pipette.
Can you explain what you're doing, Adam?
I am pipetting some sodium hydroxide into my hair samples, and that's gonna help to extract the DNA when I heat it up soon. We're here to explain to you
what we're doing. What do we have in here, Adam?
what do we have in here, you ask. Right. So the first thing we had to do this morning was pluck out all of our hairs, like billions of hairs, like it was painful.
We found that leg hairs were particularly good for this.
Yeah, I claim that discovery for myself. So you see in here, you might see a few hairs, you see. So, we've got some hairs and we like extracted the tips and put them into this,
Yeah.
call it
Well, first,
Ebendorf.
I can't remember.
Yes. And then we added sodium hydroxide.
Yeah, sodium hydroxide. That was necessary to break down the cells to help release the DNA, which is what we want.
94 degrees for 10 minutes.
On DNA, you have the sugar phosphate backbone, which is negatively charged, obviously, due to the phosphates. You know, 1 phosphate, 3 negative charges. And so we wouldn't want the hydrogen ions to just attach to it, you know, and neutralize the charge. So we want to make it alkaline, so the hydrogen ions will react with the alkaline solution and leave the sugar phosphate backbone as negative, you know?
He knows the biology. You should trust him.
We've genetically altered them so they have a human version of a particular gene called amyloid precursor protein, or APP. So we do the PCR for the APP gene. If the mice are transgenic, we'll detect the human APP, whereas if they're not transgenic, there'll be no gene in there, so the PCR won't work and we won't detect. That's too much. So what's happened is that you've taken it all the way down to the second stop. So cut that back out. And this time make sure you take it only down to the first stop when it's in.
You're on camera.
We're ready to add the second primer now. So you've done this before, haven't you? You should know what you're doing. Same again, but with the second primer. And we can get the second tube going too.
The second time I added all of this.
Daniel,
All the primers are added.
what's going on here?
Okay, right here, what's happening is we're using pipettes to add DNA primers to a solution of DNA polymerase and free nucleotides. So what this does is that mixture has the ingredients to make DNA. But DNA polymerase, which is sort of the enzyme that makes DNA copies, can't do it without these primers. These primers are like the starting sentence, like a capital letter of a sentence. We need those to start the DNA replication, and that's what they're doing. They're adding these capital letters.
One of those stations you have to work in the lab to get it. And then to run it, you use these to select. So we'll run already. And I've already programmed in the mouse program, so we just press proceed again. And then this tells us the temperature is on and the step it's on. So that's it going. Stop there. And then you want to put your comb in.
Not yet.
It's the end where the door. Excellent. Stop there. If we make it too deep. Take that glove off, but take it off inside out, like that. And then we're ready to program in the mouse. So we're going to put the comb in. And then we're going to take that glove off, but take it off inside out, like that. and then that can go there.
First contaminant.
One of the things that makes Alzheimer's disease incredibly difficult to treat is that it's a very, very slow progressive disease. So if you were to lose one brain cell, you wouldn't notice the difference. If you were to lose 2 brain cells, you wouldn't notice the difference. So you have to lose quite a lot of brain cells before you start getting symptoms of Alzheimer's disease. By the time you start getting symptoms, firstly that means you've lost a lot of brain cells. Once the brain cells are gone, we can't put them back. So we need to start treating the disease very, very early on before we get the symptoms. Only we can't predict at the moment who's going to get Alzheimer's disease. So we can't start doing that yet, which is one of the reasons it's so difficult to find a cure. You rest one hand on the side so that I've got something to support my pipette. And you take the tip in, so the tip is in the liquid. It's not all the way through the well because you don't want to stab the egg rows underneath. And then you just very gently pipette out, and the sample buffer has glycerol in it, so your sample should sink nicely to the bottom of the gel. And then again, you lift the pipette out of the well before you take the plunger up, otherwise you'll suck everything out of the well, which will cause more problems in the gel than it did in the tube.
Yeah. Doesn't seem good, but...
It's not bad. Is that this one done? So we've got to do the ladder next. Nice. So do you want to set your pipette to 10 microliters and then you can add your ladder? That was good.
Going down with this. That's it. This. Yeah, that's it.
Control number 1 is in the gel.
Okay, I'll do number 2 then.
And after this is completed, we'll run a current through, and that should hopefully separate the DNA, and we should be able to see the band of APP. Yeah, band of APP should appear. Oh, sorry. Yeah.
Good.
Nice. Excellent. Now I'm going to put these back on ice. These are ready to run. So do you want to turn that on? And I've already set it on wood. And to check that this is ready, what we do is along the wires you should see bubbles. So that tells us that we know that it's running. And then a good idea is to let it run for just a couple of minutes and then come back and have a look, just in case you put something on backwards or the wrong way round, because you want the samples to go that way through the gel and not that way, because you'll run out of the gel and be lost. Which is sounds ridiculous, but it happens eventually to everyone. So this is the negative control.
That's nothing.
That's nothing. So we added no DNA and we've got no product. And that's good because it means we didn't contaminate the reaction. So we know that we can trust the results. And then another thing I always do is I'll always add DNA from a mouse who I know is wild type and DNA from a mouse who I know is transgenic. And this is just to make sure that the PCR reaction works, because if I didn't get any products in here, I know that these work, so I know that there should be something in there. And these show us what we expect. We've got the mouse gene, which is smaller, so it's running slightly faster.
So that's what all the mice should have.
That's what all the mice should have this. And we've got, in the transgenic mouse, we've got the APP gene and the mouse gene. So from these 3 lanes, we know that our experiment is okay, and we can look at the rest of the gel to make conclusions. So it should be fairly obvious which are the transgenic mice.